Today what we are going to discuss is molecular evolution. Okay, little bit we have already discussed in the last semester when we were dealing with molecular systematics, right? Biosystematics indeed, not molecular systematics. So we are going to give you a bit more detail what the process of evolution in molecular level, that is DNA, then RNA, and then protein level, how these molecules are evolving. That is what we call normally as organic evolution, right? So how these molecules are also evolving, and how to measure this rate of evolution, and what are the significance of this evolution at DNA or RNA or protein level is. Okay, so that is what we are going to deal in this lecture. So I have divided this lecture in two parts. Okay, the first part I am going to cover today. As you know, this is a very famous statement by Theodor Dobzhansky, okay, one of the famous biologists. And what he said is, nothing in biology makes sense except in the light of evolution. So unless you know evolution, you can understand nothing. And it makes no sense at all, any kind of biological theory or any kind of research. Everything you are taking cues from the evolution. So evolution lights up the entire discipline of biology, and not just biology. Evolution of what? Evolution of computer viruses, for example, right? Any kind of thing, evolution plays a big role, right? So you have to understand the process of evolution to understand at least biology, because the word evolution in my context is all about organic evolution or the evolution of life on planet Earth. Okay? So you are taking another core subject this year, that is called Evo, right? So that evolutionary part and this evolution so much interlinked. So each course is going to complement each other. Okay? So on molecular parts I'm going to deal in this class. In this course, and for the other thing more detailed about the Darwinism or philosophical constraints of the evolution, I'm going to deal in Evo class. Okay? So, what are these evolutionary studies for? Right? Examine the process of evolution, how this evolution as a process is being conducted in nature. So that is what it's one purpose of this evolutionary studies is to examine the process of evolution, right? So what drives evolution? How this evolution is happening in the world? Understanding the mutation, gene flow, and natural selection. Well, these are some of these mechanisms by which the evolution can happen, right? So it can happen through mutation. You know what mutation is, right? The point mutation or SNPs or all different kinds of mutation, including indels, the mutations in open reading frame, all those kind of basic molecular biology. You can review it. Then gene flow. Back in last year, we were discussing in biostatistics the importance of speciation, right? How the speciation happens and what are these gene flow mechanisms. So that can be an inflow, right? That is immigration or immigration. So how these are contributing to the population genetics as such, or a gene pool as such. All these are very important factors. Then natural selection. The natural selection through the adaptation, right? As well as artificial selection. Okay? The positive and negative selections, all these are very important. Again, what? Genetic drift. So you should know what this genetic drift means, right? That we are going to brush on this in today's lecture as well, in this course. So another point is examining the history of evolution. How this organism, what we see today, have been evolved, and what are the relationship of these organisms, what we see today. That is something like back tracing the process of evolution. That is exactly how we are drawing the tree, right? To see what the relationships are. So what has evolution done in past? So you are retrospecting the process of evolution in this context. Understanding how living organisms are related and how they have changed over time. So if you calibrate, temporarily the process of evolution, that is what the context is. Is that clear? So, what the evolution is, if someone asks you to define evolution, the best and probably the most safest way to explain evolution in a genetic point of view is changing allele frequencies of a population. As simple as that. Okay, allele frequency is nothing but gene frequencies. Okay, so two or more different forms of genes are what we call it as alleles. Right? So how these frequencies are changing? Some alleles are increasing the frequency and being fixed, while some allele or some genes, normally deleterious genes or genes which are any advantages, okay, selective advantages are being thrown away from the population. Some advantages are selected. That is what we call positive selection. Okay, so that is what it is. Change allele frequencies of a population over time. Is that clear? So, well, due to 4 processes. These 4 are the core processes driving molecular evolution. Okay, mutation 1. Then selection, that could be natural selection. You know the natural selection of the Charles Darwin that we are going to illustrate in forthcoming lectures. Then the artificial selection. What is this artificial selection? It's nothing but breeding, right? Captive breeding, right? You are increasing, for example, different colors of carrot, okay, or different sizes of apple. All these things, quantitative or qualitative traits, you are improving, okay, by selective breeding. You can see a number of breeds of dogs, for example. Chihuahua is very big, tall one, right? While a Pomeranian or some small breeds are all there. How did we create it? It's all created by human being, by artificial means. It's not, what you call stochastic breeding of this one. These are all introduced breeding or captive breeding. Okay? Then, well, gene flow is a very important factor contributing in evolution as well. And then the genetic drift. Okay, genetic drift, especially when the population approaching the bottleneck situations. That is when the genetic drift is a lot more important. Okay? Well, don't worry, we are going to illustrate everything. So now here, the DNA as the genome. Mostly it's DNA, the genome, right? Especially for everyone, all life except few viruses, right? DNA is the genome. So, template for the synthesis of messenger RNA, then tRNA, of course, tRNA is also coded by the genome, then siRNA and rRNA, right? So, the DNA is a core molecule. So, that is basically the prima facie mechanisms of the evolution or the candidate for the evolution, molecular evolution. Anything happens to the DNA is what driving the evolution forward. Okay. So you should review the structure of DNA and RNA that I'm giving to you as an assignment. Simply, all these books on biochemistry and molecular biology is having all these topics, right? So structures, 2 rings, A and G are purines, and while the pyrimidines have only 1 ring, then C and T are pyrimidine, right? You should remember all those structures. Then complementarity. How these are complementarity? I mean, base pair is complementary, right? AT is, 2 hydrogen bonds, while GC is 3 hydrogen bonds. Then the base pairing, you should know how this base pair or wobble base pair happens, right? Strand synthesis, 5' to 3', that is the direction of the strand synthesis or replication, all those kind of things. Just review it when you have enough time, okay? So, the genetic code. Again, you should review that, but what I would like to illustrate here is that triplet codons are made up of 4 nucleotides. Triplet means 1, 2 and 3. Something like codons, right? Only 3 columns, 3 codes, that is exactly triplet codon means, right? So, Har Gobind Khurana, from Punjab, this is a guy prize for, discovering this triplet codon, right? Well, total possible codons is NPR, right? That is actually a permutation, right? You might be remembering your biostat days in last year, how we calculate this one, N power R, because the order matters. Here the order matters. First position, second position and third position, right? Then repetitions are allowed, because A could be 1, not just simply ATC or ATG. It could be ATD or ATC as well, right? And 4 power 364, 61 sense and 3 nonsense codons are made up of, right? Then you should know what degeneracy means. Here, you see first and second positions are all conserved. While third position, it could be U, C, A or G, right? So UC codes for PHE, that is phenylalanine, and leucine is called by A and G, right? So that is what the degeneracies. Okay, so mechanisms of the molecular evolution are mutations, as we've already discussed what mutations are in DNA. Okay, as well as in splicing mechanisms or spliceosomes. So mutations happening in spliceosomes also result in molecular evolution. Okay, so that is not well understood, these mechanisms, how spliceosomes contribute in evolution, the process of evolution. So that is a potential topic for future research, or also for your PhD research if you intend to go for PhD. Recombination is it's a natural as well as artificial recombination we can perform, right? Horizontal gene transfer, especially for viruses and bacteria, how these genes are transferring laterally. Okay, that is horizontal or laterally, not vertically. Vertically means from parent to the offspring. Okay, or first generation to the second generation, right? Forthcoming generations. But this is lateral gene transfer from one to another one. An excellent example is how eukaryotes are formed from prokaryote by endosymbiosis, right? The origin of chloroplast and mitochondria. Yeah. Then the gene duplication is a very important driving force behind the molecule. So, you should know what the ORFs are. Very simple, right? Each strand has 3 open reading frame because of triplet codon, right? In 5-3', always it is in 5-3', as transsynthesis as well as a translation. Okay? Total 6 ORF per double-stranded DNA molecule, because 3 in one direction, 3 in another direction, because this double helix or double-stranded, right? Point mutations or SNPs in third position, that is third codon position, rarely causes amino acid change. So highly degenerate, the third position of the 30% only cause change in amino acid if it mutates. Okay, third position. But in the case of second position, it always changes. If some mutation happens in the second position of the codon, it always changes the resulting amino acid. So it is non-degenerate, we can say. Okay. Now, mutations in the first position, most of the cases, that is 96% of the situations, is that mutations in the first position of the triplet codon changes the amino acid. Okay. So that kind of mutation is what we call it as non-synonymous mutation. Okay. And what synonymous mutation is, if you do this kind of mutation, and if the resulting amino acid is conserved, that is, the amino acid is not changing, so that is what you call synonymous or silent mutations, right? So synonymous or silent mutations causes no change in the resulting amino acid. Is that clear? Now non-synonymous means non-silent. Okay, so that causes change in the amino acid. Fine. So site within codon matters. 4-fold degenerate. What does that mean? CUN is an example of 4-fold degenerate. That means all mutations are synonymous. That is, this position is non-degenerate basically. Is that clear? 4-fold means any kind of, let it be A, T, G or C in the third position of the codon, the resulting amino acid is all same. Okay? So now there is something called 2-fold degenerate. In this example, UUN. So whatever this N be out of the pool of 4 amino acids, 4 nucleotide bases, it results only in 2 amino acids. Okay, 2 by one, 2 by another one. So that is what it is. Only 2 amino acids of all mutations. So that is what we call it as 2-fold degenerate. Okay? Non-degenerate, for example, NUU. So in the first means, that is what the ambiguity is, right? So if N is A, it is going to alter the final amino acid. T is going to result in yet another amino acid. G by the third amino acid and D by the fourth amino acid. Okay, so that is what non-degenerate is. Are you clear with these terms? Now mutations, as you know very well, there are 2 kinds of mutations. There is transition and transversion. The understanding is very important when we discuss about models of molecular evolution. There are several models that we are going to discuss in detail. You should know what the difference is, transition and then transversion, right? Replacement of purines with purines, so pyrimidines with pyrimidines. That is what the transitions are. Okay? So four possible transition errors: A to G, G to A, C to T, T to C. Errors in the sense mutation, right? Transition mutations can be of four types. Right? Occur when due to steric and chemical similarity. Okay, occur often because of the steric in the sense the structure of the physical structure of chemical molecules, that is the steric hindrance, you might be knowing that, right? Then the chemical similarity. Chemical in the sense, is it going to be a hydrophilic amino acid or hydrophobic amino acid? But we are not discussing about amino acids here. It's about the bases. But even bases, right? It could be having, in a simple sense, is it a two-ring structure or one-ring structure? That itself is different, right? In the case of purines and purine to purine or pyrimidine to pyrimidine, the ring structure is not hampered. So that means a high level of chemical integrity is maintained in these transitions, or four kinds of transitions. Well, the transversions of purines with pyrimidines or vice versa. So that is a lot more deleterious in an evolutionary point of view, right? 8 possible transversion errors: A to C or vice versa, A to T, G to C, and then G to T or vice versa, right? Makes it 8 total. So if mutations, that is molecular evolutions, occur randomly, suppose everything is all mutations are occurring randomly, that means that there are 8 possibilities here, right? But there are only 4 possibilities here. So chances that transversions occur is twice, right, than the probability that the transitions occur. So probability is doubled in the case of transversions, but that is not true. Okay, so transversions are actually occurring very rarely, because you must be knowing why, right? Because these are highly deleterious, right? So transversions rarely result in synonymous mutations. Almost all transversions result in non-synonymous, non-silent mutations where amino acid is changed. And even amino acids, the structural or the chemical or steric similarity of the replaced amino acid is also drastically different. So that is why the transversions occur only rarely. In the case of nature, in nature, it occurs very rarely. So you know that in nature it occurs rarely. So if you do some kind of calculations and you see is it synonymous or not synonymous or a ratio, that will give you an outlook about is it being selected. Okay, so that we are going to discuss later, probably in the next class. So how we are going to calculate or how we are going to detect the selection just by looking the transitions and transversions. Or if you know the mutations are the synonymous mutations and non-synonymous mutations, and if we do a ratio of it, we can able to detect the selection. Okay, we call as case selection. Okay, so majority of transversions render new amino acid with similar chemical properties. For example, hydrophobic amino acids are being resulted. Okay, so that the tertiary structure of the proteins are not altered much. So that is very important, right? The tertiary structure, if it's altered, then the entire function of the proteins can be entirely different, right? If the structure is also hampered. Well, mutations, transversions in non-coding positions are subjected to another constraint. So till now we are discussing about synonymous and non-synonymous. All these are coding, right? Amino acid. We are discussing about the amino acids. That means positions are coding DNA sequences of CDCs, right? CDS. Now, if it is happening in non-coding position, for example, introns or intergenic sequences. So what are the other constraints? Avoidance of long homopolymer structures, right? So if it is mutations are causing a lot of homopolymer structures, for example, T, right? 20 times T. These kind of structures are going to cause a lot of, because the polymer tends to slip through these homopolymer structures. Okay, so that is one of the problems. Then conservation of RNA tertiary structure, especially messenger RNA. The tertiary structure of this messenger RNA can be, can be altered by mutations in mRNA structure used through this kind of mutations in that molecules. Then conservation of binding sites of proteins, especially transcription factors, or even the polymerases, right? Various proteins which are involved with the DNA synthesis or expression, right, are being altered by mutations in non-coding positions as well. So that is also a very important theme in molecular evolution. Then indels are a lot more deleterious, right? Because indel is going not just causing one amino acid change, but it's going to change the downstream. Right? Everything is going to change it because the ORF, open reading frames. Right? While 3 consecutive indels in exons result in ORF conservation. Consecutive and same, right? For example, 3 insertions, that will not result in enter ORF change. Okay, so that is why if you look at that, this kind of indels, mostly additions or deletions are 3 times, so that the entire thing is not changed. Okay, so if it's changing the entire downstream, then that is going to have a huge impact on the survival of the species. Right? That is not happening mostly. Okay, so indels are rare in exons but frequent in introns. So indels are very frequent in intervening sequences. Viruses mediate indels extensively to have as many proteins possible with a small genome. Well, that is a strategy of these viruses. Because the genomes are quite small and they cannot code for a lot of proteins. But even with such a small genome, they can code as many as 6 different proteins from a small stretch of DNA by mediating these indels. So they are very smart in that case, right? Highly evolutionarily adapted for indels and for the conservation of biomolecules and resources as well, viruses, right? Mutations in spliceosomes can destroy the gene function, especially because of the alterations in expression of the genes, right? So gene cannot be expressed or it can overexpress because of the mutations in spliceosomes. And what is a spliceosome? It's a complex of small nuclear RNA and protein subunits that removes introns from transcribed pre-mRNA, right? Segment. This process is generally referred to as splicing. Right? It's very simple, but you should know how this splicing is happening. I'm not detailing in my class. But mutations in spliceosomes is another driving force for the molecular evolution. Okay? So viruses again extensively make use of spliceosome mutations. Okay, so again they are very smart in that case. So more and more research are being unclouded on how these small molecules like siRNA are having a great impact on on the molecular everything, right? Central dogma, including the expression of the genes and the control of the gene expression. Then, of course, the process of recombination and horizontal gene transfer, you should know very well about. Recombination happens in diploid eukaryotes during meiosis, right? That is between homologous chromosomes, right? The exchange of the alleles, right? Or allele shuffling is happening during, meiosis of the diploid organisms, right? Then horizontal or lateral gene transfer, which is frequent in bacteria and viruses. Origin of eukaryotes and prokaryotes, or retroviral infections results in FGT to host somatic cells, okay, somatic or vegetative cells, are being altered by retroviral infection. Retroviral, for example, what? HIV, right? So people with HIV, they are having a lot of FGT in their somatic cells, because these are retroviral infection, or hepatinavirus, that is the hepatitis B virus. That is another example of a virus possessing reverse transcriptase enzyme. Okay, so that is a DNA virus, but needs an RNA intermediate for its life history to complete. Clear? Well, another very important mechanism is the gene duplication. Duplication of the entire gene or segment of it. Okay. So it could involve single genes. So if single genes like hemoglobin is, duplicated, for example, hemoglobin and myoglobin, you see that one, or different subunits of the hemoglobin could be a resultant of gene duplication. Okay. So if it's single genes, the term is called paralogy. Okay. So paralogous genes, you must be remembering in the last year I explained what paralogous gene is and versus orthologous genes. Orthologous genes are genes separated by speciation events, while paralogous genes are genes separated by duplication events. Okay, so gene duplication and speciation, these two are entirely different phenomena. Is that clear? Then if entire chromosome duplicated, so that is what we call it as aneuploidy. Okay, so for example, Down syndrome, trisomy 21, 21st chromosome is triplicated, that is trisomy. Okay, so disomy is, well, we are a diploid organism, we have two copies. Okay, but there are some diseases like, for example, Jacob's disease, so it's caused by monosomy. We have only one copy, instead of two copies, only one copy. Trisomy is a lot more common, lots of diseases are being formed. The most famous of it is Down syndrome of 21. Okay. Then entire genomes can duplicate, that results in something called polyploidy. And this is especially common in the case of plants. So wheat, for example, or any kind of, most of the plants are polyploid, especially triploid or quadruploid. Okay, so that you can actually see that this polyploidy, another mechanism of molecular evolution, right? Complex body plans of multicellular animals made possible by molecular evolution through gene duplication of homeobox genes. Okay, HOX, Hox genes are duplicated, and each duplicated genes are having a different function. That is how this organ systems have evolved. Okay, so that is very interesting. You can read a story about Hox genes in New Scientist. Okay, that I'm going to link up in the course website. Go and have a look at. Okay, that's a very interesting story about the evolution of Hox genes. Now, coming to the population dynamics aspects of this evolution, polymorphism. You must be knowing what a polymorphism is, right? Mutations that are passed on to the offspring and then coexist with the original gene, wild type. Mutated type and wild type coexisting in a population at the same time. And these are genetically inherited. Okay, those are what you call polymorphism. That means presence of more than one form. That is polymorphism, morphological form. Poly means more than one, right? Different variants of a particular locus, that is the driving force. That is the reason why this polymorphism is happening, right? For example, in P, different of garden P. These are all resultant of presence of different alleles, okay, different polymorphic alleles of that same plant. Okay. So retroviral genome replications is highly error prone. That is one. Okay, the replication itself is quite error prone, and that error is the reason why many polymorphic genes are present in retroviral DNA. Okay, so that it's a mechanism to increase the polymorphism. So some organisms have deliberate strategies, molecular strategies to increase the polymorphism by inherent error-prone polymerases in the cellular machinery. HIV exists in host as continuously changing swarm of genomic variants. All these variants are what? Polymorphic variants, right? HIV is HIV, but in a host, for example, an AIDS patient, you are taking blood sample from various locations of his body. Then you are doing a genome sequencing to see these are all same sequence. No, right? The sequence can vary tremendously from, for example, from a blood sample or from your hair DNA sample, okay, or muscle sample. because these are all highly polymorphic. And even, are these all same species? That is the question there. Okay. So to refer these species of HIV, the term quasi-species is formed, right? It's a kind of a swarmer kind of population, quasi. Okay, it's not true species, but it's a kind of a mixture of different species, right? So diploid organisms always carry two particular locus that we know. One inherited from our father and one from our mother. Okay, recombined during the meiosis. Both are identical. So if two alleles are identical, homozygous, and if it's different, heterozygous, right? Very basic, but you should know these things, okay? Don't miss out these basics. So heterozygosity means what? Polymorphism. It's the same thing, right? Heterozygosity or polymorphism, presence of two or more different forms of the same gene, that is allele in a population. That is what the heterozygosity, the term means. Okay? Two equal and simultaneous peaks at single position in the sequence electropherogram, because we have this sequencer here. Color encoded graphs, right? So you can see two peaks, simultaneous peaks. So that is a show, that is a proof that at that position there are two or more polymorphic DNA. You can even see three peaks together. Okay? So those are how to detect the polymorphism in analysis of the sequence. So there is something called fixation rate. What is that? Fixation. Evolution, you know what evolution is, right? It is change in allele frequency of a population over time. Right? So some alleles are lost over time. So what the human beings are right now and what we were during the time of the lineage splitting from Neanderthals were bit of difference are there, right? So because some alleles that are not giving any evolutionary adaptation, evolutionary advantage are being lost, being thrown away, but some advantageous mutations are conserved. Clear? So some alleles increase frequency. Well, frequency or probabilities are always expressed between 1 and 0. 0 means nothing and 1 means 100% of one population. Okay? So if it reaches 1, that means 100%, we call that this particular allele is fixed in that population. Fixed. Okay? So rate at which this occurs, rate at which fixation of one particular allele in a population occurs, that rate is what we call rate of molecular evolution or rate of fixation. Okay? Substitution rate, mutation rate, evolution rate, all these are the same term, and this means that the rate at which one allele is fixed in a population. Are you getting the concepts? Okay, rate, that is what we call the rate of molecular evolution, right? Successive fixation of alleles. Various alleles are being fixed at the same time. Various, not good alleles are being taken out of the population, results in long-term evolution of the species. So that is how the species is evolving, by successive fixation of the alleles. Okay? So but there is yet another of the diagram which I forgot to change in this one. I'm going to change in the web version of it. Okay? So this diagram depicts, well, in a simple sense, this is the time. Okay? And this is the allele frequency. Allele frequency, it is between 0 and 1. And here it is depicted three different alleles, right? Allele 1, allele 2, and allele 3. Right? This is round, square, and this is triangular dots, right? So this round dot, you see that frequency is going down as the time increases, while these two alleles are being fixed, but it takes a long time. So once it approaches the present date, both are fixed. So that means that this particular allele is 100%. That means all individuals of that population is having two different forms of the same gene. So that two forms are these two alleles. Is that clear to you? So that is what it is. Okay? So that is what the rate of molecular evolution is. And this indeed depends upon these factors. One is generation time, That is exactly why if you want to study about evolution or molecular evolution, you have to look at phages, bacteriophages or viruses, because the generation time is very high. Or even bacteria, where the generation times you can say it in few minutes, right? 5 minutes, for example, for in the case of E. coli, right? That is why generation time is a very important factor. But if you say for human beings, for example, or any other mammals, where generation time is quite high, more in years you express it. So it's not practical to study evolution or molecular evolution in higher animals. Okay, that is why it is. Then fitness of organisms carrying polymorphism. So organisms can have polymorphism, but if they are fit in a particular environment, okay, so then that fitness confers advantage to fix that particular polymorphism in the population. Okay, so that's very important. Fitness of the organism. So positive and negative selective pressures. So that we are going to illustrate later what this positive or purifying selection is and then negative selections are. Okay. Then population size, that matters a lot. If it is a small population, then rate of molecular evolution could be quite high than a large population. Then the phenomenon of genetic drift. Okay. Then reproductive potential of individual. That is also a very important thing, right? If it is not reproductively, highly potent, then rate of evolution could be lesser, drastically lesser. Then competition of alleles. If two or more forms of the same allele, and the competition between these two alleles is yet another factor, that contributes in the rate of the evolution. Okay. So selective pressure could be, fit alleles in the population will be subjected to positive selective pressure. So alleles that is conferring some evolution advantage. Okay. So that will be subjected to positive selective pressure. Okay. So non-fit negative selective pressure. Is that clear? Non-fit alleles, that means alleles which are not giving any advantage in environment or in evolutionary context, that will be subjected to negative pressure. Means you are taking it away from the population. You don't need those alleles. Okay, it is baseless to spend resources for an allele that is not giving any advantage of a species in the population. Okay, so in the environment indeed. So positive and negative selective pressures, right? Positive means good alleles are taken out, taken in, and fixed in. Fixed means approaching 1 of the frequency of that allele. And negative selective pressure means bad alleles are taken out. Okay, as simple as that. Heterozygosity of both alleles at one locale can be advantageous. It could be even advantageous. Two different forms of the same gene at the same position. Okay, locus means the same position in the chromosome. Heterozygote is more fit than homozygote because it has got two different kinds of functions, right? So forms of functions, right? That is what polymorphic. So two different proteins, for example, coded by heterozygous. So that is actually advantageous for the survival of that species. And that is what we call it as balancing selection. Okay, you are balancing. Is it positive selection, negative selection? Here it is balancing selection because it is heterozygous. Heterozygous means more than two forms of the allele. Okay, so both are conferring some advantage for that particular individual. One good example for this is homozygous HBS allele. Okay, so HBS is hemoglobin. Okay, hemoglobin allele results in sickle cell anemia. Homozygous means HBS and HBS. If both are HBS, then that results in sickle cell anemia. But if only one allele is HBS, then another one is normal hemoglobin, then of course it's only a carrier, and the carrier is basically have more resistance to malaria. So that is why it's very interesting to read more about this sickle cell anemia and the geographical distribution of it. It's distributed in the areas where malaria is higher. So that is basically an advantage. Okay, it's a strategy of evolutionary strategy to have this heterozygous allele in those regions where malaria is more. So it confers some advantage for the infection with the malarial parasite, that is Plasmodium.